In this video, we will continue talking about the organ of Cochlea. We just want to appreciate it more. Um, the first thing that would like to state, just from the last video, is you find this among the chambers. So this organ is seated right here, okay, in red. And so we just want to familiarize ourselves with the surrounding of this organ, and really get into what is it about this organ that makes it go from mechanical, or from having a mechanical stimuli, to turning it into electrical potentials. So the organ of Cochlea, as I said, is the main organ that changes these stimulations so that we're able to perceive what it is we are hearing. And it sits on the green membrane here. If we can remind ourselves of the chambers, we have the scala tympani, and then we have the scala media and the scala vestibuli. So the green membrane is the membrane that is between the scala media and the scala tympani. So it sits on that membrane, and we also observe that it has these red components here, cells that have hairs, and these hair cells, they pierce a second membrane that is known as the reticular membrane. Okay, so the reticular membrane is another membrane that we should familiarize ourselves with. Another membrane is that over these hairs, we find that the hairs themselves then pierce, they're moved by the movement of another membrane. So this is the reticular membrane, basilar membrane. Then we have the next membrane here, which is something that we already introduced in the last one. So this is the tectorial membrane. And then over and above it, we have the vestibular membrane, which is a membrane between the scala vestibuli and the scala mediae. So the blue one here is the vestibular membrane. So four membrane that we have talked about so far. So really the structure is within the scala membrane, the scala media, I beg your pardon, is within the scala media, and it is composed of hair structures, which can also be known as auditory receptors. So these auditory receptors, they respond to movement. Okay, so once the tectorial membrane, of course, it's over them. So once they fix into, they penetrate the tectorial membrane, which covers these rows or these teeth of hairs. They embed into this membrane. Find that however the membrane moves, these hairs are also going to move accordingly. And once they move, we see that it is that mechanical movement that initiates an action potential, or a change in electrical potential. So we will see how that really happens in a bit. So we also see that this organ extends. All right, so here on the second diagram where you see green, it extends all the way from the apex to the base of the to the base of this cochlea that we had earlier talked about. So the process of the hairs go through a lamina, the first lamina, the reticular, and then the hairs are covered by viscous fluid around them in the scalar media. So the scalar media has this fluid surrounding. So this fluid surrounds, okay. So it's covered by a thin viscous fluid around. But also we have this elastic tectorial membrane that we have talked about, and the tips are embedded in it. We have pressure coming in, and when this pressure coming in, depending on the frequencies, it's going to vibrate a particular part of this membrane. And once it vibrates that particular part, then you have now the tectorial membrane moving for that particular component along the cochlea, all right? So the organs of Corti along the cochlea, you see that it starts from the base to the apex or the helical lemma. But you see that different frequencies of sound are going to they're going to influence, or they're going to vibrate different parts of this cochlea. It's more or less like a keyboard, where you start from part of the keyboard until it goes all the way to the apical area. The keyboard will start from this base all the way to a very higher note. Several other things that you must take care to know is, before we come to diagram 3, is along the scalar media we have cells. All right, we have cells which again we'll refer to soon, and these cells are able to produce a certain type of fluid for the scalar media, and this fluid, you find that it is high in potassium. It is very different from the fluid that you find in the scala tympani and the scala vestibula. So the fluid you find in the media is known as endolymph. And the fluid that you find in the other two is known as perilymph. Now, perilymph and endolymph have got different they vary in especially ionic consistencies. One thing you should know is that in the endolymph you have a high potassium level and a low sodium level, and that is very vital for the physiology of this area. So what we have in diagram 3, of course, is the tectorial membrane. And the tectorial membrane is where now we have the embedment of our hair cells. So they embed into the structure. But then we also say that there is another sort of lamina that just holds these cells, known as the reticula. Okay, so we have the reticular membrane. So the one in green is the basilar membrane, and sitting on the basilar membrane are cells. On diagram 3, we have the 3 types of cells that we're going to talk about. We have the normal cells that are sort of nutritional cells that cover the hair cells. And the hair cells, we have two types. We have the outer and the inner. And the outer are in rows of three. So you have three on the outer part, and then they go along. Okay, they go along the cochlea in this way. So you just have a line of them outside, and they're in rows in three. And then in the inner part, you just have a line of the inner cell. Before we delve into these hair cell structures, note that they have both efferents and afferents. First of all, because the outer hair cells are in threes, you find that you have over 20,000 outer hair cells, compared to something like 3,500 for the inner hair cells in each human cochlea. So the cell bodies of the sensory neurons are at the base, of course. They are at the base of each of these cells. And 95% innervate the inner cells. So, and then you have 5% innervating the outer cells. So each sensory neuron innervates several outer hair cells. So you have one that is innervating several of them. Now, in terms of efferents, you will note that most of the efferents terminate on the outer rather than the inner hair cells. So we've actually shown something here where you have the outer cells innervated by these efferent cells, efferent fibers. The afferent fibers, efferent, going to the effector, so afferent, these go through the 8th cranial nerve, okay? And you see that really the primary sensory, the ones that sense what is happening, primary sensory structures between the two are the inner hair cells. So most of the afferents, you see, they go to the outer. We'll see why. And the efferents are really coming in, afferents rather are really coming out from the inner cells and going through the 8th cranial nerve to the CNS. So coming back to the cells, the inner cells, as I said, are primarily the sensory cells, and these are there to generate the action potential, which then go through the 8th cranial nerve. The outer cells, on the other hand, they respond to sound.